Okay, good evening everybody. So welcome, it's lovely to see you here and welcome to everybody who's online as well. We all know the purpose of the meeting today and what a wonderful occasion. We're looking forward to hearing from you, Rajko. After he's given his speech, just to confirm, there will be plenty of opportunity to ask questions, but they're all in the bar upstairs, so there'll be no formal question answering here. But please do join us in the Student Union. And also Julian Marr, somebody who needs no introduction, is going to be giving the vote of thanks, but remotely. So don't get anxious that Julian isn't in the room. He is all lined up to give the vote of thanks as well. And I'd like to thank many of you for coming here today. It is lovely to be having in-person events again. So I'd like to just say a few words. We may hear a more eloquent version of your career in due course, but just for those of you that don't know, that initially your career began studying biochemistry at the University of Zagreb in Croatia, and you went on to get your master's degree at the National Institute of Medical Research in Mill Hill. I think now it's been flogged off for some luxury flats. Don't know how luxurious it was when you were there. You did your PhD at King's College London, and at that stage you were studying IgE antibodies. A glittering career of a postdoc in Cambridge, and you entered into the field of TB on your return to King's, this time with IgA. You came to St George's, your first academic appointment in 2006, and what a star you've been, and you've continued to pursue your work in TB. And you've really been very successful and part of the major £8 million euro bid to fund mucosal TB vaccination. Although that is going to be the focus of your talk, you're happy to do any number of other terrible infections and give opinions on, and you have considerable internal and external influence through a number of roles that you undertake, which we may hear about later. I have to say before inviting you to come forward how much I personally have really enjoyed getting to know you through my time at St George's. I know we haven't seen each other that much face to face, but I clearly remember meeting you very early on when I did a number of tours around. You were always lovely to me, and you also gave me a great induction and tour of the Category 3 laboratories. So the fact that you made me welcome, and also you were so eloquent and happy to share your knowledge, I know that is a facet of your personality that other people really enjoy. So it's great to have you here, and it's wonderful that you've been promoted to become a professor, and we really look forward to your inaugural lecture.
Well, thank you, Jenny, for that very kind introduction, and thank you all for coming here this evening, and thank you all those online attending my inaugural lecture. It's a real pleasure for me to give this lecture. So as you've seen from this poster, I will be talking mostly about my work in tuberculosis, specifically new approaches for prevention of TB, but I will be also touching on some other works that I've done in the past and sometimes distant past, which I hold quite important in my career. So TB doesn't need really much of an introduction, but I thought for this audience I would highlight some of the key issues to do with TB, this terrible disease that's still with us after thousands of years. So it kills a very large number of people every year. 1.5 million was the figure in 2020. And so to put it in the context, since the start of the COVID-19 pandemic, which killed 6 million people up to now, TB killed still 3 million people, so half of those people. So this is still a very, very large number of people who needlessly died from preventable and curable disease. Another big issue with TB is that, of course, so many people around the world are already exposed to this pathogen in asymptomatic form. Nevertheless, those people are at high risk at developing TB disease at some point. So this is a big problem in controlling this disease. If untreated, TB can be fatal. In fact, about 50% or more cases will die within 5 years if not treated. So this is a serious disease. And while there are treatments, they are not particularly easy. They last for a very long time, up to 6 months, and in some instances much longer still, especially if it's drug-resistant TB. Then there is, of course, that unholy alliance with HIV. As many of you know, TB and HIV tend to go together, and many HIV-infected individuals die from TB. And finally, I would like to highlight the issue of the vaccine for TB, which is called BCG. And this is a very old vaccine, still in use, but unfortunately, it is not very good, and it hasn't succeeded in bringing TB pandemic under control. So TB is a truly global disease. It affects really all of the world, but the most affected areas are the broad tropical belt around the world, specifically Sub-Saharan Africa and also Southeast Asia, bearing the brunt of TB. I mentioned treatments. Just one more slide to indicate how difficult and complicated these treatments are. So many drugs involved, duration 6 months. That's not easy on any patient. If it's drug-resistant TB, that can go 2 years or even longer. So obviously very difficult for patients, very toxic treatments, and of course very expensive treatments too. And while there are some new recent drugs on the horizon, it is yet to be seen whether they will have a major impact in improving treatments for TB. And just a bit about the vaccine, because most of my work was to do with new vaccines for TB. So the current vaccine is called BCG. It stands for Bacillus calmette-guerin. So these are the two French physicians who developed this particular bacterial strain, attenuated bacterial strain of N. bovis. And that was some 100 years ago. And this vaccine is the most heavily used vaccine in human or in medical history anyway. And so it goes to show that we still rely on it. But unfortunately, it shows very variable efficacy around the world. It's still protective against severe forms of TB, especially in infants, but it doesn't do too well in preventing onset of this asymptomatic or latent TB infection and subsequent reactivation in some of those individuals. And here I'm just illustrating why BCG is so unreliable. So these are different clinical trials performed in different geographical locations. And if you consider efficacy of a vaccine to go from 0 to 100, you can see that in some of these trials, BCG did reasonably well, but in many of them did very, very poorly and close to zero efficacy, basically. So we really need a better vaccine for TB. And my final introductory slide is just to introduce the pathogen itself. So the pathogen is Mycobacterium tuberculosis. So this is a bacterium, not a virus. And it is a very particular bacterium. It has unique features which make it very successful. One of those features is that it grows extremely slowly. And that complicates everything when it comes to research with these type of pathogens. It can be isolated from sputum of TB patients. It shows as a rod-shaped bacterium, and it typically infects and survives in the lungs, and specifically in the type of cells in the lungs called macrophages. It is transmitted by aerosol, meaning same route as SARS-CoV-2. So these are both respiratory pathogens that can be easily transmitted by aerosols. So if someone coughs, then those bigger mucus particles will probably fall down, but the fine aerosols that contain live bacteria or viruses will continue to float in the air and remain suspended in the air for long enough to be a risk for anyone in the proximity of that individual. So having just said that TB is a difficult organism, it has many unique features which makes it very difficult to work with. The question, I suppose, is why did I choose to work with TB? And I wish I could tell you that perhaps I always wanted to work with TB and that was my dream, but probably closer to the truth is an answer that a colleague of mine many years ago at King's College, when I was doing my PhD there, gave to somebody when they asked her why she was doing what she was doing. And by the way, that was nothing to do with TB. And that particular postdoc was a Chinese lady who didn't suffer silly questions and simply shot back, "I just joined the rush." And I believe that this is probably true for a lot of us. We end up doing things, going after opportunities rather than meaning to work on something forever. And so that's really pretty much what happened to me, but you'll hear about it a bit later. But where did I start my career, and how did I get into TB? So I obviously wasn't born in this country. I was born in the country called Yugoslavia, and I did my undergraduate degree at the University of Zagreb at the Department of Biochemistry. That's my university badge. And I also did my master's degree here in collaboration with the National Institute for Medical Research in London. And then I started doing my PhD here too. But unfortunately, things got a bit complicated then for me. And around that time, a civil war broke in that country, which had several federal units and they couldn't agree on anything. So that was a difficult situation to be in, because I was born here in Bosnia and grew up there, but I'm an ethnic Serb and Serbs live in Serbia. Yet I found myself studying and living in Croatia, and this war was about Croats versus Serbs versus Bosnians versus Croats and so on. Everybody was fighting somebody. So my situation became really complicated, difficult, and I was beginning to think, what to do. I had to make some hard choices, and to put it simply, the choice was either to join the war effort or try and get out of that situation and try and pursue my interest in science. So I spent a lot of time in this particular cafe called Amadeus, named after a basketball player whose nickname was Amadeus, thinking about my future and what I should do. And this is really where I made that choice, and particularly after some terrible personal losses in that war, that I would remove myself from that situation and I would go somewhere else and pursue my career in science. And I needed a way out, and fortunately for me, the way out was provided by this wonderful lady, Professor Hannah Gould, at King's College London, Drury Lane campus, who offered me to do a PhD in her lab. Not only did she secure a PhD studentship for me, she also sent me an advanced payment so I could afford one-way ticket from Zagreb on one of the very last flights that was allowed to leave Zagreb for London. And this is how I got to London and really then enjoyed my time working with Hannah. So this is also my first adventure into conventional immunology. I worked there on a molecule called CD23, which is a receptor for IgE, antibody, IgA antibody, of course, being implicated in allergies. And what we found there was that this receptor does also many other things beyond that primary role. Specifically, it synergizes with other molecules on the surface of B cells. So these are the cells that make antibodies, very important immune cells, so that the threshold for activation of those cells is significantly reduced via antigen receptor signaling. So that was really nice, and I enjoyed that. And I wanted to do more work in B cell biology and with antibodies. So I then moved to Cambridge to join a renowned American immunologist, Douglas Fearon, who was also interested in B cell biology, in the Clinical School of Medicine in Cambridge. And there we did some really nice work, which resulted in some really important findings in terms of B cell development. So these B cells, which you can isolate from your lymph nodes, from the general centers within your lymph nodes, these cells tend to proliferate and grow or expand. This is what we call clonal expansion, and you can actually measure that. You can see their numbers are increasing. However, with the right cytokine environment, these cells stop growing, stop proliferating, and instead begin to produce antibodies. So they become antibody-producing cells, plasma cells. What we found is that if we transduce these cells with this particular molecule called BCL6, which is a transcription factor, then these cells never stop growing, even upon stimulation with these cytokines, and they can never produce antibodies. So basically, they've been forced to grow continuously. And so what that means is that BCL6 molecule is the major regulator of B cell differentiation from that clonal expansion stage to terminal differentiation into antibody-making cells. And in addition to that, this finding also explained the occurrence of certain types of large B cell lymphomas, which are due to dysregulated BCL6. And so that finding turned out to be very important for understanding the development of B cells, and today, in fact, remains one of my most cited publications with something over 300 citations in the literature. But at that point, I started also my young family in Cambridge, and for personal reasons, me and my partner wanted to return to London. However, I knew I wanted to stay with immunology, albeit I would have preferred to go into applied immunology, so something like infectious diseases. And I looked for opportunities to do so. But having spent 3 years in Cambridge, I thought I couldn't go anywhere higher. However, I found myself on top of Guy's Tower on floor 28, where this gentleman here, who I'm immensely proud and happy to see in the audience, invited me to join his group and work on a project to study the role of mucosal immunity for protection against tuberculosis. So this is really where I met TB first, and specifically to look into the role of antibodies against TB. So there was a dogma that antibodies can't do much against intracellular pathogens such as TB, but that view was not necessarily shared with everyone and needed to be challenged. So we started working on that, and we made a lot of progress demonstrating that antibodies, specifically IgA antibodies more so than IgG antibodies, could indeed suppress TB infection in a mouse model of infection. So as you can see here, mice can be infected with TB either intranasally or by aerosol, and then if they are treated by antibodies, that infection is reduced by more than tenfold on this logarithmic scale. So it's a substantial reduction. Not only does the antibody reduce the infection, but it also stops the relapse of infection upon incomplete treatment. So in collaboration with Francesco Dieli in Italy, we demonstrated that if you infect mice with TB, of course it grows. If you treat mice with TB drugs, and they can be treated just as humans, in this case isoniazid and rifampicin, then that goes very low. However, if you leave those mice, infection will come back, it will relapse. But with IgA antibody treatment, it stays low, clearly indicating a potential that this could be used as some sort of adjunct treatment with TB drugs. Juraj and I did a lot of work with this, but it was a hard work because there was still a lot of reluctance among TB community to accept that evidence. And that was reflected in many grant applications being turned down and even papers bouncing from journal to journal. So we wrote a review in Lancet to basically make a case that this topic should be now given due attention, that there is definitely potential in this and it should be explored further. But my problem with this was that at Guy's, I did not have animal facilities to do TB work, and that really meant that I had to rely on collaborations to do that kind of stuff. So I was beginning to think of where I might go to be able to do animal TB work. And so I then ended up, or rather visited this Starbucks cafe in front of Guy's Hospital. This is the second cafe in this tale of two cafes, so with much less pompous name. I went there because I went there every morning. I would take my young children to a nursery nearby. By that time I'd be exhausted. I would go to the cafe and have my coffee and read the Times and just recover a little bit before going to work. So that's what I did that morning when I noticed that someone approached my table and slightly annoyed me. I was beginning to be slightly annoyed who was invading my personal space and time. When I looked up, I saw this gentleman there, and without saying as much as hello, he just simply asked, "How would you like to work at St. George's?" I said, "Yeah, that'd be very cool," and that was really that. So that's how I came to St. George's. And at St. George's, I finally had what I needed to do this work. St. George's had a wonderful animal facility that included also this highly specialized lab for TB work. This is a picture I took a few years ago. There is me helping one of the PhD students. It had everything that I needed to do TB work with animals. So I wanted to continue working on this IgA antibody, but of course, if we were ever going to use it in humans, it needed to be made human antibody, not a mouse monoclonal antibody. So we collaborated with Professor Jenny Wolf at Dundee and made indeed a human version of that antibody. And then I demonstrated that even the human version was still protective in mice. Albeit this time mice were a bit modified, so they also expressed human IgA receptors, so that the receptor and the antibody were matched in terms of species. And not only it reduced the infection, but it also very much improved lung immunopathology. So lung immunopathology in TB is really important because this is really what causes disease and eventually loss of function of the organ. So infected hosts tend to have extensive granuloma infiltration, so that their lungs become granulose. But the treated mice had much clearer lungs, so that can be quantitated. So there was much better immunological picture of the lungs. So I was then thinking, where could we most realistically use this treatment? I mean, obviously there are several scenarios. TB has got lots of different manifestations too. And to my mind, the clear way forward is to test whether it can work against multi-drug resistant TB, because that's where the greatest need is for, coming up with some new treatments that could shorten that very long two-year long treatment or improve cure rates. So we did a lot of studies with that, and a couple of PhD students in succession, first Jill and then Andy, did a lot of experiments, and the summary of that work is shown here. So what we see here is that if you apply this treatment to these animals, you get a very significant reduction of the bacterial load in their lungs, about 50-fold reduction compared to untreated mice. And then depending on when the treatment is initiated, you may lose some of that effect, but assuringly, even when the treatment is initiated during a very well-established infection, it still has a significant impact. So clearly demonstrating that you could use it in people who are already infected with MDR-TB. If we were going to use it in humans, it would have to be through aerosol route. So we wanted to deliver that treatment directly to the lungs, not to the blood. So the antibody would need to be aerosolized, and so we needed to see whether it can survive aerosolization. And so Andy again spent a lot of time doing that. So we use this type of nebulizer here, and when we nebulize the antibody, we very quickly realize that we are losing a lot of material. Antibodies are very complex structures. They don't like to be bashed about by very violent forces during the nebulization. However, Andy tested several additives and found conditions that could completely recover protein from the nebulizer, and not only recover the protein, but protein that is still functionally active. So we haven't lost functionality of the antibody either. And all of these additives that Andy used were permissive for human application. So at that time, last year, unfortunately, our lovely animal house that I introduced a bit earlier closed down due to some difficulties, and that created a number of problems for me because so much of my work depended on animal studies. So then I started collaborating with Professor Galit Alter at the Harvard Medical School. I sent her some of our 2E9 antibody. She collected also a panel of anti-TB antibodies from all around the world that have been reported to have some protective potential against TB. Some of these antibodies were targeted to secreted proteins of TB, others to cell wall proteins, others yet to intracellular molecules such as ours, and so on. And I was very pleased to see that, in that head-to-head comparison, our antibody indicated here in red performed really well in terms of protection against TB. In fact, it was one of the most protective antibodies here. Galit also showed very nicely that some of these antibodies seem to induce metabolic programming within the macrophage, which leads to better bactericidal response, which is very much in line with our working hypothesis all along, that targeting through IgA receptors will increase bactericidal state of macrophages. So that work is really exciting and is currently being reviewed in the journal Science. So to conclude this part, I'm very happy to report that this dogma that lasted for many, many years, that antibodies can't play a role in intracellular bacterial infections, is now finally debunked. And to illustrate that, very recently, the last 5-6 years, there's been a spate of high-profile publications, including from Galit Alter herself, in top journals, all reporting that antibodies play a role in different circumstances, different scenarios, but all supportive of the role of antibodies in TB. So moving on to now my interest in mucosal vaccines. So I started work on TB vaccines as soon as I joined St. George's, but really this gathered momentum about 6-7 years ago when we managed to assemble a large consortium that included 14 countries from Europe and Africa. And we managed to secure €8 million funding from the European Union to allow us to investigate a large number of mucosal TB vaccine candidates. And we here at St. George's coordinated that project. But first of all, why could mucosal TB vaccine work? Why do we think so? Well, here is an incomplete list of some of the indicators why it should work. For example, there are precedents. There are several mucosal vaccines that have been licensed and work very well, including those for polio, cholera, influenza, and so on. Also, mucosal vaccination can induce both systemic and mucosal immunity, which is something which systemic immunization doesn't do too well. It only induces systemic immunity. Mucosal vaccination is also amenable for large immunization campaigns. There is no needles involved, which is a safety thing, because there is no transmission of other blood pathogens such as hepatitis B and HIV. If these needles are reused, and they are in some resource-poor settings. But despite these progresses and advantages, there are still, of course, a number of challenges to be resolved, which explains why the mucosal vaccines haven't really made as much progress as we hoped. In the case of TB, the most realistic mucosal route would be respiratory delivery, so that we can match the route of vaccine to the route of infection. We believe that that will induce the strongest, the most protective immune response at the site where infection takes place. And we also believe that inhaled TB vaccine would be best suited as a boost to current BCG vaccine, which is injectable vaccine. Simply because so many people have been already vaccinated with BCG, we can't ignore them, so we should work with BCG rather than try to replace it. Okay, so our approach to do this study was can be represented by an inverted pyramid. So we started with many mucosal vaccine formulations. We tested them in the standard mouse model, and St. George's was the hub for testing all of those vaccines. And then we progressively moved on to more advanced models, including guinea pigs and non-human primates, of course, down selecting the number of candidates at each step. And in all of our studies, we use BCG at the baseline, and then intranasally or mucosally vaccinated animals with new candidates, and then we infected them with TB, and then did the measurements of bacterial loads in the lung and other parameters. So this is a summary of some of that work. So we tested 36 different vaccine formulations, 6 of which passed our internal scoring criteria, which included not only whether they protect against TB, but whether there may be toxicity issues of feasibility or scalability, and so on. And so we came up in the end with 3 formulations, which are shown here. They all performed very well in mice. So you can see here, this is basically amount of infection in the lungs. Blue is non-immunized mice, green is BCG immunized mice, so significant reduction, but never sterility. You cannot achieve sterility in this experimental model. And then in red is our vaccines, which again conferred further protection above BCG only. So following some more studies, we decided on this particular vaccine candidate to be our lead candidate, and it's called Spore FP1. So what is Spore FP1? It's a formulation that contains 2 components. One is the vaccine delivery system, and this is represented by Bacillus subtilis spores. Sounds a bit counterintuitive to use bacteria against bacteria, but these spores are non-pathogenic. These are harmless organisms that can be found in environment. They also live in the gut of mammals, and they have some interesting adjuvantic properties, so they can be used as vaccine carriers or delivery systems. And then the antigen being the second component, so we used 3 antigens from TB, which we made into a single fusion protein, so they represent different stages of TB infection. And the final antigen, HBHA, was there also to facilitate interaction of our vaccine with the mucosal surfaces. So the first thing we wanted to see whether following intranasal administration in mice, we can actually deliver that formulation to the lungs, to the lower lungs, where it needs to go. And as you can see from these images here, we can, you can see here that even after 48 hours, you can still identify fluorescent spores in alveolar spaces. That thing is shown here as well. And then not only we can identify them in the lungs, but they can also be taken up by lung immune cells, and that's also important, so they can be then presented to lymphocytes. This is the most important outcome in these studies, essentially whether the vaccine protects against TB infection. So we did a lot of experiments, and this is just a couple of experiments showing again that unvaccinated animals had high bacterial loads in their lungs. BCG does what BCG does, which is a 10-fold reduction, and nobody knows why that cannot be pushed any further. But our vaccine, our mucosal vaccine, induced further reduction in bacterial load in the lungs and also to some extent in the spleen. So this study was led by a supremely talented young immunologist in my group called Alistair Copeland, who also did extensive immunological evaluation of these immune responses. So we saw, for example, that systemically we could see a production of two most important cytokines in terms of protection against TB, interferon gamma and IL-17A, as indicated by these bars. So while they are not correlates of protection, they are prerequisites or indicators of protection, so it's good to have them there. But also we saw a pool of different types of memory T cells, which are also important for long-term protection. As you can see here, all these red bars, which is the vaccine-induced T cells, are higher than other controls. But really the most importantly was demonstration that we can induce a local immune response in the lungs. And so this is represented by a number of immunological parameters, but probably most importantly by something called resident memory T cells. So these cells, they migrate to the lungs or any other organ indeed, and then they stay there. They don't go back into circulation unlike the conventional cells. So they stay in the organs and they can respond very rapidly upon pathogenic challenge. So it's really good to have those cells at the sites where infection might occur. So what we saw here is that our vaccination induced a significant increase in these resident memory T cells, as you can see from these numbers, both in the CD4 and CD8 compartment, and significantly more than BCG alone, so a lot more resident memory T cells. We were then able to confirm the data from the mouse model also in the guinea pig model. So this was done in correlation with Karolinska Institute. Again, our vaccine, as boxed here, induced a significant reduction of the bacterial load above BCG alone, and in the spleens actually it demonstrated sterility. We saw a lot of animals having no infection whatsoever in the spleen, which was never seen before in these type of studies. And also we could see that the vaccine reduced significantly lung immunopathology. So normally infected animals have heavy immunopathology, lots of granulomas, as you can see here, but immunized animals had much clearer lungs. So the final model to test this vaccine was the non-human primates. Now this is obviously a very advanced model, very difficult, expensive, and there are of course ethical issues and all these things. But it is a very relevant model for human TB. So for non-human primates, we had to deliver our vaccine by aerosol. There was no choice anymore. We couldn't be injecting fluid into their noses. So we aerosolized spore FP1, and so again we used Omron nebulizer, and this is how it works. You place the fluid inside, you activate the device, and then that aerosol comes out, you can then collect it. Either it goes into a host, or you can collect it in a vessel like this, and eventually you see condensation there. So, but with non-human primates, it's a bit more complicated because you can't tell animals to breathe in or breathe out. So they have to be placed under masks, they have to be anesthetized as well, and then aerosol is delivered over a period of time. And of course, because they are not hermetically closed, they breathe in, breathe out, there are losses. So we were aware of these things, but we couldn't really, there were no easy technological solutions to mitigate against them. Nevertheless, we went ahead and we did our best, and we immunized those monkeys first with BCG systemically, then by aerosol, by spore-active vaccine. And so first, let me show you some of the immunological parameters from this study. So we saw here, for example, that our vaccine induced a sharp increase in antibody responses in the blood of these animals to all of our antigens, and also different type of antibodies, IgGs and IgAs. Really, the most exciting bit was this, the mucosal vaccination induced mucosal IgA antibodies. So these were antibodies recovered from lung washing or lung lavage from these animals. We also saw strong cellular responses in the spleen. So here I'm showing interferon gamma, which is clearly higher than for the BCG only immunized animals. So that all sounded and looked really good. Unfortunately, we fell a bit short when it came to the most critical outcomes from that study, which is the survival and disease progression, and also lung pathology. So in terms of survival, we saw a small trend towards improved survival for the vaccinated group, but this was not statistically significant, nor was the study powered to have that outcome as the primary outcome. There were only 6 animals per group. But in terms of lung pathology, which is the most reliable readout in this particular experimental model, we saw that both vaccine groups had significantly reduced pathology compared to unvaccinated animals, but unfortunately there was no difference between BCG alone and BCG plus spore FP1. So that meant that we had to think about how to proceed forward, and obviously we had 2 animal models that showed very encouraging data, and 1 animal model, which by the way is a very problematic model, not just for this vaccine, but in general, that there was no protection there. So I'm currently, in fact, receiving consultancy paid by Bill Gates Foundation to do what they call gap analysis and identify what we need to do, whether it's to repeat another non-human primate study or whether to circumvent it altogether and go to clinical trial to see how we can take this particular vaccine candidate forward. So that's TB. I will stay with mycobacteria for a short while, but switching now to another disease, which is also mycobacterial infection, but very unlike TB. So this disease is called Buruli ulcer. It's caused by a bacterium called Mycobacterium ulcerans. But unlike TB, Mycobacterium ulcerans doesn't infect lungs, it instead infects skin. It affects skin forming lesions. Initially, those lesions then progress to ulceration, and ulceration progresses to basically eat the skin from the whole limb or any other part of the body. So this can be really looking awful. I deliberately chosen a case which is not too bad, but this can lead, even though the mortality from this is low, it can really lead to high morbidity and stigma and poor quality of life. This is one of those neglected tropical diseases, so there's not awful lot of interest in them. It's endemic mostly to some parts of Africa, and there are about 50,000 cases a year, so nothing compared to TB, but nevertheless a significant problem for those affected by this disease. So we teamed up with a research institution in Ghana, and Ghana had a significant number of cases of Buruli ulcer. And so one of the clinical scientists, Dr. Justice Bokai up here, came to St. George's to do PhD with me, and Justice is sitting at the back there. And so we wanted to establish an animal model of Buruli ulcer to see then whether we can develop vaccines against it. And indeed, we were successful in developing a very nice, reliable animal model. So you inject Mycobacterium ulcerans into the foot pad of the mouse. After a few days, it swells. After a few days, it begins to ulcerate, but we would usually stop our experiment before that happens for humane reasons. As you can see here, that swollen foot is much bigger than the other one. And you can actually then measure that swelling, and that's the indicator of progression of the disease. We then made several vaccine formulations, and in particular, our hopes were with this particular formulation called Borulivac, which had 3 or 4 different components in it. I'm not going to go into details, but it is a cellular vaccine. There are no live cells in there, and it's protein-based, and also mycolaphrone toxin from ulcerans itself. So when we immunized these mice and then challenged them with the pathogen, what we saw is that some of these vaccines had some effect. Obviously, this is unvaccinated group, so all animals developed heavy swelling. But our Borulivac formulation completely prevented swelling in these foot pads. And if you present that result in a different way, as percent population protected from swelling, whether it's small swelling or large swelling or any swelling, again, you see that our vaccine formulation completely prevented swelling in all animals. So it seems to work very, very well. And we are currently writing a paper, but going further, we are aware, of course, that this is a neglected disease. We are aware that pharmaceutical industry probably won't be too interested to invest a huge amount of money to develop a vaccine that will sell only 50,000 doses. So that's an issue, and we were aware of that issue all along. But nevertheless, we are happy that we came up with something that seemed to work very well, and obviously will not file WHO about that. And if there is ever need to take that forward, it's there, and it could potentially be taken forward. And in the final part of my presentation, I'm going to talk about some ongoing work, which is to do with COVID-19. So we all work a little bit about COVID-19 these days. And so our interest really is to develop an inhaled COVID-19 vaccine. Because we believe that having localized immunity in the lungs and upper respiratory tract will be more beneficial than having antibodies somewhere else that need to migrate and somehow find the virus in the respiratory tract. So our main approach is to develop this, what we call self-adjuvanting vaccines. So that concept is based on the fact that we are basically within the structure of the vaccine, including all that it needs to induce an immune response without the need for exogenous adjuvants. So that vaccine had 3 components in it: cholera toxin B as a molecular adjuvant, obviously SARS-CoV-2 antigen, and a portion of IgG antibody. And each this part has a different role to play in this vaccine. So that vaccine can polymerize. It looks something like this. And I must say that I'm being fortunate that in our lab we have a supremely talented molecular biologist, Dr. Myung Kim, who could make all these very complicated molecules. So this particular vaccine formulation, which we call COV-PCF, which we submitted also a patent for, we propose will work by engaging immune cells in the submucosa, so those cells which underlie epithelial layers. And then they will take up the vaccine, produce antibodies, including IgA antibodies, which will then return into the lumen. And there they can intercept virus before it had enough time to propagate so that it can cause severe symptoms or even transmit to the new host. So So again, importantly, we needed to see that it can be aerosolized. We use the same nebulizer, and here we show that indeed it can be nebulized, protein fully recovered, and then not only recovered, but it retains full activity when it comes to those two components in terms of their binding to their target receptors. So ganglioside for chlorotoxin B, or RGG receptors on antigen-presenting cells. As you can see here, no difference before or after aerosolization. Now we are doing immunization as we speak. We've just started doing it at London School of Hygiene, so we haven't got data yet to show. But what we do have is very encouraging data using exactly the same vaccine approach, same vaccine platform, just different pathogen, in this case dengue. And we did a lot of work with these self-adjuvanting vaccines for dengue. And indeed we saw in the context of dengue that this vaccination approach induces a very robust antibody response in the blood systemically, but more importantly, it induces a very robust immune response also in the mucosal fluids, in the lung washings, represented by IgA antibodies. So this is very exciting. I think if you can replicate that with the SARS-CoV-2 vaccine, that's exactly what we are after. And then, of course, we want to see that vaccine, those antibodies will be also neutralizing. And just finally, the other thing we did was to see, because we plan to use this vaccine as a boost, and I know that this word boost is not very popular at the moment, but as an inhaled boost, and because to those who have already been vaccinated or infected, so those individuals will have antibodies against SARS-CoV-2 present. We wanted to see what our vaccine interaction with those antibodies might mean, how vaccine might fare then in terms of antigen, vaccine uptake by the immune cells and amplification of the immune response. And indeed, what we find is that serum from immunized individuals, SARS-CoV-2 immunized individuals, does complex with our vaccine, whereas serum from non-immunized animals does not. So we think this is a good thing for our vaccine. We think that this is going to form even larger immune complexes with a greater chance of antigen uptake by antigen-presenting cells. So this would be something of a boosting at antigen-presenting level beyond the concept of just expanding the existing immunological cell memory. And as I said, we are doing immunizations at London School, again, because we can't do it, unfortunately, at St. George's, and we are very excited to be able to test their neutralizing potential in the next couple of months or so. So this is my final slide. I think that's the story so far, really. I mean, I can't say that I have cured any disease or cured any patient, but I'd like to believe that these contributions will help us achieve those objectives sometime in the future. I'm particularly proud and pleased that we have managed to overturn this long-standing dogma about the role of antibodies in TB, which now is a very fertile ground for research, and it really is, I believe, down to perseverance of myself and Uri, who stuck with this topic for many years, that we are revisiting this topic now. We think that our antibody-based, IgA antibody immuno-based therapy could be used for adjunctive treatment of MDR-TB. We would like to take this forward. And there is a real opportunity to advance mucosal vaccines against diseases such as TB and COVID, and including to human clinical trials, and these are my main objectives in the years to come. So current preclinical evidence is encouraging, but there are challenges ahead, and of course they will have to be overcome, including the pharmaceutical industry, for example, is currently poorly set up for inhaled vaccines. Only a very, very small company, I personally know only of Glaxo Wellcome, that are interested or develop technologies for developing inhaled vaccines. So finally, just I would like to say that, however, these incremental progresses that we make now will be critical to fully harnessing, I believe, the potential of mucosal vaccinations and therapies in the future. couldn't do this work on my own, of course. There are lots of people I would like to acknowledge, people from the past and people now, and in particular my former mentors back in Zagreb and also at King's College, Hannah and Juraj. Many wonderful collaborators I had over the years, and I still collaborate with many of them. And then, of course, the wonderful people at St. George's who were incredibly helpful to me in my career here at St. George's. And I'm pleased to see that quite a few of them are in the audience. And of course, I need to thank all the funding agencies who supported my work. And last but not least, my family and friends who supported me in my efforts. And finally, thank you all for your attention.
Julian's meant to be talking now, but his camera refused it.
Oh, there you go. He's coming. He's coming.
One moment.
I've been told to wait for my picture to appear on the screen, which is almost as scary as turning up behind somebody at the cafe, I think. Can I go?
You're good to go, Julian.
Thank you, John. Well, first thing to say is thank you so much to Rajko for a real tour de force of an inaugural lecture. Fantastic to hear the story of your research from before I knew you to now, and what a story it's been. I think what really comes through is your dedication and your passion for the topics that you research, but clearly you're not afraid to challenge preconceived dogma, and I think that's one of the most important personality traits a scientist can have. I first met Rajko when he joined Juraj's research group at Guy's back in 2000, and as Jenny said, he came with a very impressive reputation. My first impression was of a very serious young scientist who was very knowledgeable, read a lot, and was very up to date. Slightly scary, actually, but that may have had something to do with his military training, I think. So, as you have seen, my first impressions have been borne out. He is a very serious scientist. He is very knowledgeable, and yes, he can still be slightly scary. But what I've also discovered since then has been his humor, his faithful friendship, and that deep vein of thoughtfulness which runs through everything he does. Contrary to Rajko's story, I don't go around cafe bars sneaking up behind postdocs, but I did take the first opportunity to bring him to St. George's. And it's been no surprise to me that he has made this wonderful career progression to senior lecturer, then to reader, and now to full professor. As he has established and developed his research interests and his own research group at St. George's, his emphasis on identifying and helping to mentor young scientists and postdocs has been clear. His approach to building a strong research team is based around, I think, the 3 C's: collegiateness, camaraderie, and coffee. He works really hard to develop and establish collaborations, both locally and internationally. And of course, he's been an excellent citizen and contributor to St. George's in research and beyond. I can't help thinking that recruiting Rajko to St. George's has been great for St. George's. But in turn, coming to St. George's was also a great move for Rajko. The university has given him tremendous support and space to develop independently. And that it has also provided that collegiate environment in which he's been able to build lifelong collaborations. So that really is something for us to be proud about too. I know that Rajko will go on to bigger and greater achievements, and we will follow his work with great interest. But for the meantime, I invite you to join me one more time to congratulate him and to thank him for his wonderful presentation this evening.